So I'm going to talk to you today about some research done by some folks in my lab groups. This is a segment of some students that I worked with when we were at a conference a couple years ago, or about a year and a half ago. And in particular, the project I'm going to talk to you about today was done by Thomas and Olayinka and Laura Neveling and Angela Pozo-Ramajo. Angela is a postdoc from Spain who was working in the lab. Lauren and Thomason were both students who graduated in 2007. Lauren is now working in the pharmaceutical industry, and Thomason has actually gone on to nursing school. So before talking about cataracts, I'm going to actually make an attempt to talk about the structure of the eye, and sort of keep in mind this is a chemist talking about the structure of an eye. So many of you likely know more about the structure of the eye than I do. So I'm going to focus on the piece of the eye that I worry about the most, which is this thing here, which is the lens. Okay? And the lens is what captures the light, okay, as it comes into your eye, and helps to focus the light so that it can impinge on the retina, which is connected to the optic nerve, which is going to send the signal to your brain. So the lens, it basically is a lens. It's what it sounds like. It's capturing the light, it's focusing the light. And if you think about it, to be a lens, there's one major thing that a lens needs to do to be effective. You need to be able to see through it, right? It needs to be transparent. That if you can't see through the lens, if light can't get through the lens, that's a problem. So the whole idea of the lens, or the cellular structure of the lens is quite interesting because it's a cellular structure that's designed to maximize transparency. So this is a picture of a lens. So this is an actual human lens to give you an idea of what the size looks like. And it looks frighteningly like a contact lens in some way, except it's not. This is real tissue. And if we think about what the lens is made up of, it's a fascinating organ in the sense of the central part of the lens. In order to be transparent, these are cells which are really nothing but bags of protein. They've gotten rid of everything that could scatter light or that could prevent light from getting through. So there's no nucleus to the cell. Because there's no nucleus, they obviously don't have their own DNA. They can't divide. They can't undergo mitosis and all of those things. It's really just a bag of proteins. Now, in these cells that are in the center part of the nucleus, and so these are the ones that definitely are just bags of proteins called crystallins, and I'll show you the crystallins in a minute. Because they don't divide, these are not cells that are going to turn over. You're not going to get new lens nucleus cells, which means that the cells of the nucleus of the lens, the central part of your lens, are the same cells that you've had there basically since you were in utero. Okay? They have to last a really long time. Okay? And that's going to be important when we talk about cataract formation later on. Okay? That these are the proteins that are made there are proteins which have to last basically, if you live 100 years, 100 years in good shape. Now, if you take proteins, if you take egg whites, for example, if you break an egg, you put egg whites on your counter. Egg whites are basically also just a kind of glob of protein. And you let it sit there for 100 years. Okay? Probably 50 days into that 100 years, the egg whites are going to start to look kind of gross. And they're going to look gross for a few reasons. Things are going to grow on them. Okay? But even if you could keep them fairly sterile, the proteins in egg whites, are going to start to fall apart over time. Okay? The proteins in your lens can't do that. They have to be very stable, because they're sitting there, and they're sitting there at body temperature for a long time. And it's important for their function not to fall apart. So a major protein of the lens nucleus is this protein called gamma-D-crystallin. It's got a really cool shape, or I think it's a really cool shape. It's in what's called a Greek key shape. It's very stable. So here is a protein that you can produce if you can manage to keep it sterile, if you can keep things from growing on it. You can let this sit on your kitchen counter, and this will be happy in this shape for an extremely long time. It's a very, very stable protein. And its main job is you have lots and lots of these crystalline proteins. They pack together in such a way that they maintain the transparency of the lens, and they help to focus the light. Now, cataract is a disease that really is a clouding of the lens. And so if this is a healthy lens, this is a lens that, this is a cataract lens. And basically, this clouding of the lens is the accumulation of misfolded protein which has begun to unfold, become to get tangled up or aggregate. And those aggregates of protein actually lead to a clouding of the lens. And physiologically, so this is what normal vision would look like, and this would be vision through a cataract lens. Okay? It's like you're looking through a thin film of sort of clumps of protein that have accumulated within the lens. One of the more interesting cataract patients in history, fascinating case of cataracts, is Monet. Monet actually developed cataracts late in his life, actually had cataracts in both eyes, and it was sort of, I think at the time, actually a quite experimental cataract surgery that I believe he had quite late in life, in his 70s. Here's a picture of the Japanese painting of the Japanese bridge that Monet did early in his career, and here's a painting of the Japanese bridge that Monet did during a period sort of after he had developed cataracts, before he had cataract surgery. Okay? And you can see that there's actually an interesting change in the view and an expression, in his expression, of the difference of what he was seeing. So the goal of the work, so what Thomason and Lauren and Angela were working on, was to try to come up with a model system for testing different hypotheses of what causes cataracts. So there certainly is a sense that over time, if you let a protein sit around long enough, sooner or later it's going to begin to unfold, and so sooner or later you should get some accumulation of these misfolded proteins. But there are other hypotheses that certain environmental stimuli, for example, ultraviolet light, other types of environmental changes, might actually trigger effects of the crystallines at an earlier stage, younger, for younger patients, which lead to formation of cataracts earlier on. And so we're interested in trying to see, could we get a model system for testing some of these hypotheses, of what are some of the things which can lead to the unfolding of the protein and the formation of these tangles. And the hope is that as we learn more about these molecular mechanisms, we could suggest some targets for drug development. Now, this is going to be a bit of an unsatisfying presentation. I can warn you now. I should have warned you maybe 15 minutes ago. That I do not know the answer to any of these things. So this is very much work in progress and problems that we're actively engaged in. And this is the goal. But just as warning, there's no answer coming along. Now, what one challenge is we want to study this protein. You can get these proteins from, we want to study the human protein, we could get it from the lens of human eyes. It's probably not a good idea for us to accumulate large amounts of this protein, and I'm not sure, I love undergraduate researchers, but I'm not sure I want to give them scalpels and patients to collect protein. So instead, what we do is we have a system for expressing the human crystalline in bacteria. So we have the gene that encodes for human gamma D crystalline. That gene has been cloned and placed into bacteria, E. coli bacteria, and we can grow up bats of these E. coli bacteria that express the crystalline protein. Then we isolate the protein from the bacteria, and we can make large quantities of this in the laboratory. The downside is that these flasks smell quite a bit, though Angela told me that she got used to it after a while. But it's an efficient way for us to get this human protein in large quantities so we can study it in the lab. And so then the second challenge, so then we can get lots of the protein, how do we actually induce it to form cataract? Well, if, as I said, this is stable, if you just under normal conditions let it sit on the bench, it takes many, many years for this to happen. Most students like to graduate before this would occur, so we need some mechanism to speed it up. And what we found is that you can incubate this, if you add a little bit of acid, you can basically speed up the process of cataract formation, and it occurs on the order of hours instead of years. So this is a solution before incubation. This is a solution after incubation. And you can see the clouding, which is the aggregation of a protein. Lauren, who was a student in lab who first did this, got very excited. It said cataracts in a tube. So we can basically have a process for developing or following the aggregation of this protein in the laboratory, and then doing further experiments to try to dissect and understand the molecular mechanisms of what's going on. So we can make the protein, we can generate its aggregation. The final step that we need to do is to come up with a technique for understanding the molecular structure that's happening. And doing this, we do a technique that's called infrared spectroscopy.